Okay, we're moving on to our final part of Unit 4, Area of Study 2. There's a few things involved in this, but really what we're looking at is how biological knowledge, particularly DNA, is used in society and in scientific studies. So today we're going to look at these first two dot points, which is looking at a few examples of applying DNA knowledge to different cases, and then also looking at genetically modified organisms. So the four things that we're going to cover today are called gene cloning, which we've actually kind of looked at the science behind that already, genetic screening, DNA profiling, and genetically modified organisms. And the study design also specifies that you need to understand some of the social and ethical implications involved in these things. So gene cloning, notice I've got this in yellow because this is a type of scientific study, involves cloning or making exact copies of genes using vectors. Okay, and a really common example of this is the bacterial transformation that we looked at in the last lesson. But it can also be done with viruses. What this is used for, there's a few different ways it can be used. So the first is to create copies of genes that can be studied to help us understand genetic disorders. So really important in terms of our scientific understanding of diseases and disorders and things like that. It can, and this is a more new use of it, but it can be used to create copies of kind of healthy genes or fully functional genes that could be inserted into the genome of a patient to treat genetic diseases. It can be also used to mass produce specific proteins. So remembering obviously that genes code for proteins. If we can clone genes and then switch on the transcription and translation of those genes, so that gene expression, we can produce proteins. And a really good example of that is insulin for people with diabetes that don't produce their own insulin. This is what we've looked at in the last lesson, which is the bacterial transformation. So that's when a plasmid is used as a vector. That gene that we want to clone is inserted into that vector, and then the bacteria take up those plasmids and then can either just be used to divide, so we're getting lots of copies of the gene, or as we said, it could then be used to produce the proteins as well. It's the same process, I guess, with viruses. It's just that the virus is used instead of a plasmid. Okay, so it's a different vector, which would still get, taken up by a cell and then used to reproduce that gene. There are social and ethical implications of these. I'm not going to spend too much time going through these, but you definitely should have a few notes written down because it has come up before in exams. So obviously the benefits of being able to do things like this is that we can, treat certain diseases, which can decrease the pressure on hospitals and save money. Obviously, by treating diseases, we can save lives and improve people's quality of life. By using things like bacteria for gene cloning, it can reduce the need to study on animals, which is, what used to be very, very prevalent is using animals like rats and monkeys and things like that to study things. And, by saving lives, we can increase population sizes, which can be a benefit to the economy. But you'll see that's also a risk in a second as well. Some of the risks is obviously anything that's interfering with genetic variation can be risky. We've talked about how important diversity and genetic variation is for survival. So if we interfere with those things, it could impact on natural selection. It can lead to inequality. So these kinds of technologies are expensive. They're not available to everyone, and they're not available in all countries. So it can lead to inequalities. There's this big ethical debate about, is this kind of playing God or, interfering too much with natural processes? Obviously, certain diseases get prioritized over others. So that, often comes down to funding and what kind of things people, care more about and are willing to put more money into. And then I guess the other side of this point here is that it can result in overpopulation, which can put huge strain on resources, and again can be negative for the economy if we've got lots of people that require healthcare and all of those kinds of things. All right, genetic screening is basically using DNA to study whether a person has genetic differences, or maybe they're more susceptible or prone to particular diseases. You don't need to know too much about how genetic screening occurs, but just that it involves actually looking at a person's genetic sequence. This is most commonly performed during pregnancy, particularly if parents are the potential carriers of certain diseases. So you would have looked at this in year 11. Parents can be carriers, particularly of recessive alleles. So they may not themselves have a disease, but if two carrier parents, have children, there's kind of a 1 in 4 chance, depending on the recessive dominant trait of that, but there is a chance that that child then will inherit both of those alleles and have a higher chance of disease. So it's particularly used, to screen for certain diseases, but it can also be used in adults. So anyone, can have their DNA screened. Again, it's very expensive. It's only really used if there seems to be a high risk. Again, there's benefits and there's risks. Being able to identify the risk of certain diseases can increase treatment opportunities or kind of early treatment. For, really serious, genetic issues, it can allow parents to make informed decisions surrounding whether they, would like to keep their child or whether they would like to terminate. And it can be used again just in study, so looking at those patterns of prevalence of different genetic conditions, which gives us more and more understanding. Again, risks quite similar to the last ones. It's not available to everyone. It is very expensive. It's often used, to screen for diseases that don't have any treatment. So there's a bit of an ethical debate as to, well, if something can't be treated, do you want to know if there's a risk of that? Again, it ties in with this termination thing, and that obviously is a very personal thing for people about where they sit in terms of the ethics around that. Some screening might pick up, a risk of a child developing a disease much later in their life. So there's an ethical debate about, should children be told about those kinds of things? Is that something they should be aware of if there's a high risk of certain diseases, or is that just going to cause unnecessary stress? And, this is a very controversial one, but, the technology means that this could potentially be used to screen employees. So, people might, this doesn't generally happen now, but it could be used that people have to undergo genetic screening in order to get a job, to make sure that they're going to be healthy and aren't going to cost money to the company, or people might be asked to undergo genetic screening to check if they're eligible for things like health insurance or life insurance. So definitely some big ethical issues around that. Right, we're going to spend a little bit of time now looking at DNA profiling. This is a pretty cool thing, and people often are quite interested in this. But this is using DNA to basically compare or to identify individuals. So the two ways we kind of look at it really are using it in forensics. So, if you watch crime shows, they always screen for blood and body products so they can get the cells and extract DNA and try and identify who that individual is. It's also used for paternity testing. Okay, so to check if someone is the biological father of a child. A little bit of science behind this that you do need to know is that in our DNA, we have these non-coding sections. So remember we talked, at the start of Unit 3 about coding and non-coding sections of DNA. In these non-coding areas that don't determine, they don't produce proteins or anything like that, they're just there. There are these repeating sequences that are called short tandem repeats. Okay, so they appear in DNA. So let's say, these bits that are colored are short tandem repeats. They're just repeats of the same sequences. But the important thing is different people have different numbers of these repeats. Okay, so it's a little bit small and hard to see, but this is showing, 4 different people here. And the first person has 8 repeats of this particular sequence. Person 2 and 3 have 7 repeats of this particular sequence. Person 4 has 10 repeats of this particular sequence. If those sequences are compared at enough different points in the DNA, so this is just looking at one particular area, if we looked at this on, say, 10 of those, you get a very unique genetic footprint. Okay, so when you're looking at lots of them, no 2 people are going to have, or maybe 1 in 5 million will have the same number of repeats. So if we've got different, this person has 7 repeats of this certain sequence. This person down here has 12 of the first one and then 14 of the second one. What that does, and where it's useful in terms of what we're learning, is it means that people have DNA of different lengths. Okay, so we're not going in and looking at all the actual bases, but looking at the length of DNA for particular alleles or particular genes. So hopefully you're starting to think now, if we're trying to compare DNA based on its length, what type of tool we might use for that. But basically, the DNA samples are taken from individuals. Then they are amplified or multiplied using PCR. So we've got lots and lots of copies to work with. And then those copies are compared using the gel electrophoresis. Remember, that allows us to sort DNA based on size. So you can compare the DNA based on its size. I'm going to go through a few examples here because it can take a bit of practice to get your head around these things. So let's look at an example now with forensic testing. So we've got, this is the DNA that was extracted from a crime scene. And, the police have got 3 suspects and they're going to compare the DNA with the DNA of these suspects. If they know that this DNA at the crime scene was only from the suspect, so they've removed DNA that might have come from, say, the victim, or let's say there was no victim, it was a break-in and there was only 1 bit of DNA that was found, or 1 type of DNA that was found. If it's a match with the suspect, it needs to match each of these parts. Okay, so let's say these are different alleles or different sections of genes. For it to be a match, it needs to match completely with the suspect. So, if we compare with suspect 1, like these genes match in length, these ones match in length, these bottom ones match in length, but this one there's no match for. And then this, this suspect has DNA that there was none in the crime scene DNA. Okay, so that is not a match. Suspect 1 is not a match because there's some similarities, but it's not exact. If you move on to suspect 2 now, every single allele is a match. Okay, there's no parts in the crime scene DNA that don't match with suspect 2. There's no parts of suspect 2's DNA for these particular genes that don't match with the crime scene. Okay, so it needs to be an exact match. When we're looking at this, keep in mind as well that that is going to be using multiple of those short tandem repeat sites. Okay, so if we again look at this, is shown in a very different way. Generally, if you're asked a question, it's going to be something like this. It's going to show you the gel electrophoresis run. But if we look at this, we've got some crime scene DNA and the suspect's DNA. For this first section of DNA with the first short tandem repeat site, it's a match. Okay, so there's the same number of repeats for this. You move on to site 2 though, and it is not a match. Okay, so the crime scene DNA had less of those short tandem repeats than the particular suspect. So in this case, you could not say that that is the same person, because it's likely that you'll get certain sites will be the same, but all of them need to match. All right, here's another question. This was in a previous exam. So a young person wants to find out more about their genetic ancestry. They send a sample of cells obtained from a swab in the mouth, which is often how that's done, to a lab. When the lab receives that, it treats the cells to release the DNA. And then it's just asking, what's the process used to produce many copies of this DNA? So that would be our polymerase chain reaction. Makes lots of copies. Okay, and then what's often done with these kind of ancestry investigations is that they will compare the DNA of the person that is asking for it to be tested with other family groups, often with the same surname or that they think would be related. Okay, so in this question, it's, it asked which family is most closely related to this individual. So if you start at the top, that top section of DNA is actually a match with all of the families. Okay, then in the middle here, family Y and Z have DNA lengths that aren't seen in family X or in Ben, the person that's being tested. Then Ben has an allele here that actually doesn't have a match with any of those families. So that's a unique allele to him or to his family. Family X has one that doesn't match Ben, but does match family Y. If we move further down, at this point, that allele matches with families X and Y. This matches with families X, Y, and Z. Again, family X has an allele here that doesn't really match with anyone. That's quite unique. Then down here, we have some matches as well, not with this, but with families Y and Z. So basically, you want to look at how many matches there are with each of these families. So the closest would actually be family Y, because here we've got some that doesn't match. Here we've got another one that doesn't match. The only thing that doesn't really match between Ben and family Y is that he's got this longer allele, a shorter allele here, sorry, and this family has the longer allele. Okay, but the rest of them do match. So you would say that that's probably the family that he's most closely related to or was most recently related to. Okay, when we look at paternity testing, it's a little bit different, because generally the way this works is you get the DNA of a child, and then you want to find out, this is the question mark, is who's the daddy? But we know who the mother is. So with the child's DNA, remember the child, let's say, we're looking at particular alleles. The child will get one allele from its mother and one from its father. Okay, so for each of the child's DNA bands here, it needs to match, each one needs to match either the mother or the father. Or sometimes, it will have the same one. It'll inherit the same one from both parents. Okay, so again, you have to work through each of these. These are generally easier on paper because you can draw on them. But let's say this first allele, it obviously received from its mother. It's a match with that. Okay, this second allele here, there is no match with the mother. It does match with this individual. Okay, the next allele down here, it probably got from its mum. This allele is a match with that father or that person. Okay, this last one actually matches with both the mother and this person. Okay, so what this tells us is that this could be the father. Okay, you would obviously need much more evidence than just a couple of alleles, but there's nothing in this to suggest that it is not the father, because each of these child's alleles either matches its mother or this man. Okay, so it could be that person. Don't worry about, the father having some that don't match to the child and the mother having some that don't match to the child. That doesn't matter because, the mother contributed an egg that only had half of her DNA, and the father contributed a sperm that only had half of his DNA. So he's going to have some alleles that the child doesn't have, and so will the mother. All right, another example of this, a bit more complex. Like I said, you couldn't just use one little bit of DNA. Lots of alleles would be tested. So here we're going to look at 3 different DNA sites. Here we've got the M is the mother, C is the child, and then we've got 3 possible fathers. Okay, so we're going to compare for each of these sites. So we'll start with at site A. Okay, so this first DNA, this first allele here matches with the mother, also matches with father 1 and father 2, doesn't match with father 2, sorry, but that doesn't matter at this point because this could have been received from the mum. So we can't rule out father 2 yet. Okay, we'll move down to this one. Now there is no match with the mother at this point. So this allele had to come from the dad. Okay, fathers 2 and 3 are matches for this allele. Father 1 is not. Okay, so it can't be father 1 because this allele didn't come from the mum, so it had to come from the dad, and father 1 doesn't have that allele. So you could actually probably eliminate father 1. So 2 and 3 are still in the running. We'll move on to site B. This allele here, again, this could have come from the mum. We've gotten rid of father 1 at this point. We'll move down to this allele. Okay, there's no match for the mum here, so we are looking at this allele. At this point, the only match for that is father 2. Father 3 and father 1 don't have alleles that match that particular one. So we're thinking it's father 2, but we'll just double check using the last probe. First allele, again, that came from mum, so we don't care too much about that one. This allele, there was a match with father 1, but we already ruled him out all the way back here. And it does match with individual number 2. Okay, so again, we can't say 100% for sure that it is father 2, but we can say 1 and 3 are not the father. Okay, so it could be person 2 could be the father, but persons 1 and 3 cannot be this child's father. Another way we could look at this is comparing, we've got the mum, we've got 2 of her children, both of these children are that mother's children, and then the possible father. I don't really like the term alleged there, but is this the father of these children? So remember, the children are going to have unique DNA to each other. So let's go child 1 and child 2, and we're comparing, is this person the father? All right, child 1 and child 2, they got this allele from their mum, so we don't really care about that one. Child 1, this allele could have come from dad. So far, yes, we've got a match here. Child 2, this allele could have come from dad. Yeah, from this person. So that's a potential match. Let's move down now to child 2. This allele matches with mum, so we don't have to worry about it. Child 1, they have this allele here. No match with mum. No match with dad, with this person. Okay, so for child 1, we've got one allele that doesn't match either of those parents. All right, child 2, this allele matches with that father. So yep, that's another bit of evidence that that is that person's father. Child 2, this allele came from mum, so we don't need to worry about that. Child 1, this allele came from mum. Child 2, this allele is a match with the allele from that person, so another potential there. Child 1, we've got another allele here, doesn't match with the mum, and there's no allele of the same length in the dad. So there's another one here where we don't think it came from this person, or it didn't come from this father. Again, that came from mum. So at this point, we'd be saying child 1 is probably not that male's child. But we'll keep checking for child 2. Child 2, that's a match. So we got another tick. Child 1, this one did match, but you know, that's not enough evidence. There's a few that don't match at all. Child 2, this allele came from mum. Child 2, that's a match with that male. Child 2, both of these alleles can be accounted for by the mother. Okay, so what we would say from this evidence is child 1 is not this man's biological child. Child 2 could be. Okay, it's, you know, it's fairly likely. There's a lot of matches and there's no unexplained things, but child 1 is not the child of that man. Okay, moving on now, it's a bit of a change from what we were looking at to genetically modified organisms. You've probably heard of these, you know, they do get talked about a bit, but genetic modification is basically the science, it's sometimes called genetic engineering, which involves altering or changing an organism's genome. Okay, so basically it is changing the DNA of an organism. This is usually done in order to give desirable traits to an organism. Generally, we see this in farming with crops and vegetables and things like that. It can be done on other things, okay? So there are, you know, if you want to Google glowfish, you know, you can buy fish that glow because a gene that is taken from jellyfish that glow has been inserted into goldfish to make them glow, okay? So it can be done in other things. Obviously, again, lots of ethical issues around that. There's 3 main ways that you could modify or edit the genes of an organism. One is that you could insert desired genes. Okay, so you add some genes in or add a gene into its DNA. You could remove undesired genes. Okay, so if we have a gene that we know is damaging or faulty or something like that, that could be removed. Or you could alter genes. So, you know, if there's just a couple of point mutations that are incorrect or are causing some kind of damage, you could alter, you could replace a few of those nucleotides. Really important to note that this is different to artificial selection. In the last area of study, we looked at artificial selection where, you know, certain crops are bred or certain animals are bred because they've got desired traits. That's different. That doesn't involve changing the DNA. It's just, you know, choosing desirable organisms to breed, whereas this is actually changing DNA. There's 2 terms, and it's quite easy to get them confused, and it is really important you know the difference between these. Okay, so genetically modified organisms are any organisms that have had their DNA changed. Okay, if their DNA has been manipulated in any way, that's a genetically modified organism. Organisms can be altered or modified using the DNA from another species. So if that is the case, we call it a transgenic organism. So trans, you know, kind of meaning across or different, and it's the genes from a different species. Okay, so genetically modified organisms are basically what we said before. You can have DNA removed, you can have nucleotides removed or changed, you can have DNA inserted. If that DNA is inserted from another species, it's called a transgenic organism. Okay, if the DNA is inserted from the same species or it's artificial DNA, that is just a normal genetically modified organism. I know this is a bit confusing. We'll look at a couple of examples, but a good way to think of it is all TGOs, a transgenic organism, all TGOs are GMOs because they've been modified, but they're also transgenic because they're modified with another species. Not all GMOs are transgenic, okay? Some GMOs are not modified using DNA from other species. All right, three particular examples. I've used these because these are ones that come up quite a lot in the exam. And like I said, it's usually done in agriculture. One way that this can be really beneficial is that you can modify organisms or modify crops to make them resistant to insects or herbicides. Okay, so they're less likely to get destroyed by insects or chemicals that are used on crops. Okay, so an example of this is called BT corn, which is these things here. So you can see this corn here has been eaten up by insects. It can't be sold. It's not useful anymore. So BT corn are corn that have been given genes from bacteria. Okay, so bacteria are a different species to corn. And that means that those genes allow the corn to produce a protein that kills the insects. Okay, so this one here is the modified corn that is resistant now to insects because it produces this protein that came or is able to do that because of genes from a bacteria. In the quiz, you'll have to answer whether that's transgenic or not. Crops can be modified to increase their nutritional value. This can be really, really important in especially low-income countries where there's not a lot of food security. So an example of this is golden rice. Golden rice has been given the genes from daffodil flowers and also certain bacteria, again, different species, that allow it to produce vitamin A. Okay, so that makes it a more nutritious rice with more benefit for people that eat it. Another common example, and this is one of the first commercially available genetically modified organisms, is called the flavor saver tomato. There's genes in tomato that cause it to ripen. Okay, so ripening is obviously good because it makes fruit and vegetables edible, but if they ripen too quickly, they can rot. So the flavor saver tomato had the gene, the normally occurring gene that causes ripening was removed so that ripening proteins aren't produced, meaning a longer shelf life. Okay, so rather than tomatoes rotting a day or two after ripening, you might be able to keep them for a week now. Okay, so all that happened is that the gene that's naturally occurring in that tomato got removed. Okay, so in the quiz you'll have to answer whether each of these are genetically modified, transgenic, both, just one. All right, the last thing is again looking at social and ethical implications. Again, this has come up. This is generally where these come up in the exam, is to do with genetically modified organisms. So firstly, the benefits. It can make more productive crops. So it can be really beneficial for farmers. It can be really beneficial for consumers. And again, like I said, in lower income countries, it can be really, really important that no crops are getting wasted. Because it's more productive, it can mean that there's less habitat loss in farming. So, farming can be done in a more efficient way. As I mentioned, it can improve nutritional value, which is really important for health. It can improve food security. So, everyone has availability of nutritious food. Obviously, it's better for farmers if it's more efficient and profitable. They're not losing, crops from insects and herbicides. It's good for us, means better quality food often food that looks really nice, which people seem to care about. But there are also risks. So again, it's decreasing that genetic variation. Okay, so, we don't know if you remove a gene for ripening, that might also remove the gene that protects these tomatoes from certain pathogens. And so, if there is a viral outbreak, that could mean that entire crop gets destroyed. So the more we mess around with DNA, the less we know about what we're doing. These are expensive technologies generally, which again can create inequality. So only, maybe some farmers can afford it, and then those farmers are now making more money because it's more profitable and they're not losing crops. It can really lead to big inequalities. Again, a lot of people view these things as unnatural and that, they don't like things, particularly with things that we eat, that kind of unknown of modifying naturally occurring things. And then You know, if you've got two farms next to each other, and you know, this farm has genetically modified crops, this one has organic crops, so they're not modified at all, you know, cross-pollination can very easily contaminate these organic crops with these modified crops. And that can actually be really important. You know, organic certification is very strict. So for a farmer to say that their products are organic, if some of these, you know, crops ended up in here, that can affect that. Farmers can even be sued if it looks like, you know, they're using these genetically modified crops that they haven't paid for when actually it just occurred through cross-pollination. So again, worth having a few of these written down as they have come up in the exam before. Okay, so that's it for today. Gene cloning, genetic screening, DNA profiling, and genetically modified organisms. Thanks.